The topic is digestion. This lesson includes definition of digestion, types of digestion, digestion of carbohydrates, digestion of proteins, and digestion of lipids. The food we consume consists of carbohydrates, proteins, fats, vitamins, minerals, and water. Out of these carbohydrates, proteins, and fats, they release energy upon oxidation. The energy released is stored in the form of high energy phosphate bonds called ATP. Carbohydrates are the major source of energy. They produce about 50 to 60% of the energy, of the total energy. And fats provide 20 to 30% of energy, and the remaining is by proteins. Proteins provide less energy, but they are supply essential amino acids, and these amino acids are utilized in the synthesis of other proteins which are essential for various activities. Fats provide essential fatty acids in addition to energy. Water, vitamins, and minerals are vital for various biochemical reactions. Vitamins act as coenzymes. Minerals are cofactors and structural elements. Water is the medium in which various biochemical reactions takes place. All these nutrients are needed in proper proportions. If there is any deficiency of these nutrients, it leads to disorders. For example, malnutrition of protein leads to Kwashiorkor disease. Malnutrition leads proteins and energy leads to marasmus disease. Deficiency of vitamin A leads to night blindness. Deficiency of vitamin D leads to rickets and osteomalacia. And deficiency of iron leads to anemia. So our diet should contain all these nutrients in proper proportions. The diet which supplies all the nutrients is called balanced diet. Carbohydrates, proteins, and fats, they are complex macromolecules. They cannot be absorbed in the form as we consume. So the process in which natural complex food materials are converted into absorbable simpler forms is called digestion. So the chemical bonds are present between these complex food substances. These are broken down in the presence of water. This process is called hydrolysis. The enzymes which help in hydrolysis reaction are called hydrolases. Digestion occurs with the help of hydrolases. Hence, this reaction is also called as enzymatic hydrolysis. So here we can see these are the complex food particles upon digestion with the help of hydrolysis. These are formed into simpler forms. Carbohydrates upon digestion, they break down into glucose, monosaccharides, glucose, fructose, and galactose. Proteins are broken down into amino acids, fats into fatty acids, and glycerol. Now let us see about types of digestion. Based on the occurrence of the digestion, whether it takes place in the cell or outside the cell, digestion is of two types: intracellular digestion and extracellular digestion. First, intracellular digestion. This digestion occurs within the cell. The food is ingested with the help of food vacuoles which are present in the cell. We can see here. So this food is ingested. These are the food particles. These food particles are taken by the vacuoles, food vacuole. These are the food vacuoles. And lysosomes are the cell organelles which contain acid hydrolases enzymes. Acid hydrolases include proteases, carbohydrates, nucleases, lipases, etc. And these enzymes, they break down the food content present in the food vacuoles. After digestion, the digested particles are absorbed into the cytoplasm and the undigested food remains in the vacuoles. These vacuoles with the undigested food material, they are called as residual vacuoles. These residual vacuoles reach the cell membrane and the contents are removed out of the vacuole by exocytosis. They are eliminated by exocytosis. So this type of digestion occurs in unicellular organisms like amoeba, paramecium, and in multicellular organisms like sponges and hydra, and also in higher organisms in the phagocytic cells of animals. Phagocytic animal cells. And extracellular digestion. This digestion occurs outside the cell in the either in the gastrovascular cavity or alimentary canal. The digestive enzymes are secreted into the lumen of the alimentary canal and the enzymes break down the food particles present in the lumen into simpler molecules. Those simpler molecules are absorbed into the wall of the alimentary canal and the undigested food is removed out by mouth or anus. So this extracellular digestion is seen in mostly higher animals. Extracellular digestion has some advantages. This evolutionarily, in evolutionary point of view, it has some advantages. It allows to take great variety of food particles which are larger than the cells itself. And this extracellular digestion has facilitated evolution of specialized cells through differentiation and division of labor. This is the digestive system of man. It starts with mouth and anus. This is the mouth opening. This is the buccal cavity, esophagus, stomach, intestine, small intestine, large intestine, rectum and anus. The digestive glands include salivary glands, liver and pancreas. So the digestive system performs 6 different functions. It can be divided into 6 different functions. One is ingestion. Ingestion is taking food into the mouth opening. And next, secretion. Secretion is the digestive system. It produces and releases some acids, buffers and enzymes. Next is propulsion. Propulsion is the food is pushed forward through peristaltic movements. Digestion is a breakdown of the food by mechanical or chemical methods. Mechanical and both chemical and mechanical methods. Mechanical is chewing of the food and chemical is by the action of enzymes. Absorption is taking the nutrients and water into the bloodstream, first in the cells of the alimentary canal and from there to the bloodstream. And last is egestion. Egestion is expelling undigested food through anus. Now let us see how these complex macromolecules like carbohydrates, proteins and fats are digested. First, about carbohydrates. Carbohydrates include starches, sugars and glycogen. Most of the food we take consists of cereals. Cereals contain starch, that rice, wheat, millets, ragi, jowar, they contain starches, and vegetables like tubers, they also contain much amount of starch. And sugars are also present in our diet, regular diet, like sucrose and fructose, and milk sugar. Milk also contains sugar called lactose. So these starches are polymers of glucose units joined by glycosidic bonds. Starch is stored in higher plants in the form of amylose and amylopectin. So we can see here this is a glycosidic bond. This is a sugar molecule, and this is another sugar molecule, and these molecules are joined together by glycosidic bond, and the bond here is alpha 1,4 glycosidic bond. So this is amylose sugar present in the starch, amylose present in the starch, and each unit, it consists of many units, monomers, and all these are joined by alpha 1,4 glycosidic bonds. And this is amylopectin. Amylose is a simple unbranched polysaccharide, and it is helicoid in nature. Whereas amylopectin, it is complex and it is branched, and the main branch consists of alpha 1,4 glycosidic bonds, and the side chains the main chain is joined to the side chain with the help of alpha 1,6 glycosidic bonds. So glycogen is the animal reserve food, and the animal carbohydrate is stored in the form of glycogen, and sucrose and lactose are the disaccharides, and all these carbohydrates are digested by a group of enzymes called carbohydrates. Now let us see the structure of this is the glycogen. Glycogen is also having the alpha 1,4 linkages, and this is sucrose, disaccharide sucrose, and the bond here is alpha 1 and beta. Here 1,2 bond is there, and here this is the lactose, and the glycosidic bond is between beta 1 and 4. So these are the monosaccharides. This is the glucose. Here the numbering goes like this. This is the 6 carbon sugar. This is glucose, fructose, and galactose. Now let us see how the carbohydrates are digested in various parts of the digestive system. First, digestion in oral cavity. The food we consume is chewed with the help of the teeth present in the buccal cavity. As soon as the food enters into the oral cavity, salivary glands secrete saliva, and saliva consists of large amount of water, nearly 9.5% of water is there, and 0.5% of electrolytes are present, and the other is mucus, and some proteins or mucus is present, lysosome and amylase enzyme. And water present in the saliva moistens the food and it facilitates swallowing. The mucus present in saliva lubricates the food particles. It is slippery in nature. The lysosome has antibacterial action, and it lyses the cell walls of the bacteria. Saliva consists of an enzyme called salivary amylase. It is also called as ptyalin enzyme. Starch and glycogen, they are acted upon by salivary enzymes, salivary amylase enzyme, in the presence of chloride ions, and the pH required for this reaction is 6.8. So this is almost near to neutral. So starch and glycogen in the presence of salivary amylase enzyme, with the help of chloride ions, they are broken down into maltose, maltotriose, and limit dextrins. Maltose is a disaccharide maltotriose is it consists of 3 units, 3 monomers, and limit dextrins they are limited to 8 units. So next, digestion in stomach. About 5% of the carbohydrates are digested in oral cavity, and the partially such food is enters into the stomach. But carbohydrate digestion will not take place in stomach due to absence of amylase enzyme. Gastric juice is secreted by the glands present in the wall of the stomach. The food mixes with gastric juice. The smooth muscles present in the stomach help in churning of the food. By peristaltic movements, the food is churned properly. The gastric juice consists of high amounts of hydrochloric acid, and it is acidic in nature. Gastric juice, hence it is acidic in nature. HCl helps in breaking of the extracellular matrix that is present in plant and animal tissues. With this, the further digestion takes place easily. Hydrochloric acid also neutralizes the salivary amylase which is present in the swallowed food. So the softened and food mixed with acid present in the stomach is now called as chyme. This chyme, acidic chyme, it enters into the next part of the digestive system, that is intestine, through a small pore present between stomach and first part of the intestine called duodenum. So this pore is regulated by a pyloric sphincter. As soon as the food enters into the intestine, several juices are secreted. Pancreas secretes pancreatic juice, bile secretes bile juice, and the intestinal wall secretes intestinal juice, and all these juices are released into the intestine. So pancreas secretes pancreatic juice, and this pancreatic juice is alkaline in nature. It consists of 2 enzymes, alpha glucosidase and pancreatic amylase enzyme. Sodium bicarbonate present in pancreatic juice, it acts as buffer and converts the acid chyme into alkaline. Bile juice neutralizes the acid chyme to alkaline broth. So the remaining starches which are present, only 5% is digested in the oral cavity, and the remaining amylose and amylopectin sugar starches which are present, they are acted upon by pancreatic amylase and alpha glucosidase enzymes, and this reaction occurs in the neutral medium. pH required is 7, and they are hydrolyzed to maltose, maltotriose and dextrins. Maltose is disaccharide, triose with 3 monomers, and dextrins. Here we can see this is the starch molecule, and here the bonds are cleaved. Pancreatic alpha amylase is breaking down the starch molecule into maltose, and isomaltose. This is isomaltose, and this is maltotriose, trisaccharide, and some dextrins. Okay, some dextrins. The Lieberkühn follicles of intestinal epithelium, they secrete intestinal juice, and that is called as succus entericus. So this succus entericus consists of some disaccharidases enzymes called disaccharidases. They are maltase, alpha dextrinase, lactase and sucrase. All these enzymes act in alkaline medium, that is pH 7 to 8. Maltose and maltotriose, they are hydrolyzed by maltase enzyme and form into glucose. Dextrins are hydrolyzed to glucose with the help of alpha dextrinase enzyme. pH is 7 to 8. And lactose sugar is converted to glucose and galactose with the help of lactase enzyme. pH is 7 to 8. And sucrose is hydrolyzed to glucose and fructose with the help of sucrase enzyme. All these reactions occur in the pH 7 to 8, that is alkaline medium. So at the end of the carbohydrate digestion, glucose, galactose and fructose are formed. That is, the polysaccharides are broken down into monosaccharides, namely glucose, galactose and fructose. Now let us see about the protein digestion. So our diet consists of proteins also. We consume proteins, both animal and plant proteins. So our body requires 0.8 mg of protein per kg of body weight. So proteins are the polymers of amino acids and they are joined by peptide bonds. So here we can see this is a protein, amino acids, these are the amino acids and they are joined by peptide bond. This is a peptide bond. So proteins are digested by a group of enzymes called proteases or proteolytic enzymes. Proteolytic enzymes are of two types according to their mode of action. They are endopeptidases and exopeptidases. Endopeptidases cleave the polypeptide at the interior. So this is enzyme is called as endopeptidase. Whereas so this polypeptide is cleaved at this point and is broken into two chains. And exopeptidase, it exerts its action at the end of the polypeptide, at the terminal end of the polypeptide. And this exopeptidases are of two types, aminopeptidases and carboxypeptidases. Aminopeptidases cleave the terminal amino acid at the amino end of the chain. This is aminopeptidase. It cleaves at the amino end of the chain, terminal amino acid. And the carboxypeptidase cleaves the terminal amino acid at the carboxyl group. So now let us see how the proteins are digested in various different parts of the digestive system. Proteins are not digested in the mouth due to the absence of proteases in the saliva. So oral cavity consists of salivary gland. It secretes saliva, but saliva does not contain any protein digesting enzymes. Digestion, next digestion in stomach. So the protein digestion is initiated in stomach region. Stomach releases gastric juice. Gastric juice contains hydrochloric acid and two proteases, namely pepsin and renin. So this pepsin and renin are secreted first in the form of inactive forms called pepsinogen and prorenin. These inactive forms are also called as zymogens. These zymogens ought to be activated first in order to exert their action. So the HCl present in the gastric juice kills bacteria and activates pepsinogen. Hydrochloric acid and pepsin, then it can auto-activate the remaining pepsinogen into pepsin. HCl also transforms the tertiary and secondary structure of the proteins and opens the peptide bonds so that pepsin will act upon them very easily. Pepsin is an endopeptidase enzyme and it breaks the bonds at the amino end of some amino acids like trypsin, tryptophan, phenylalanine and leucine. So here, let us see, this is the pepsinogen, zymosin in active form. This is activated in the presence of HCl. It transforms into pepsin. This is active form. When soon after some pepsin is formed, this pepsin autocatalyzes, it undergoes autocatalytic reaction and transforms the remaining pepsinogen into active pepsin. This pepsin acts on the proteins and breaks down them into peptides and polypeptides, and this reaction occurs in the pH 1.5 to 2. So the pepsin, it breaks the bonds at the amino end of tyrosine, tryptophan, phenylalanine and leucine amino acids. The second enzyme is prorenin. It is also a zymosin and it is activated in the by the pepsid enzyme. This reaction takes place at the pH 2. So it is prorenin is transformed to renin. Renin is the active form, and renin is the it is also called chymosin, and it is present in infants and calves and helps in digestion of milk. So this enzyme, it acts on the milk protein, that is casein, and transforms it into para-caseinate. So in the presence of calcium ions, the para-caseinate is it undergoes coagulation or precipitation reaction and calcium para-caseinate is formed. So this reaction is also called curdling of milk. After the formation of calcium para-caseinate, pepsin will act on this calcium para-caseinate and it will transform it into it hydrolyzes calcium para-caseinate into polypeptides and peptides. Now digestion in intestine. So partial digestion of proteins occurs in the stomach region, and this food enters into the intestine, the first part of the intestine, that is duodenum. Soon after entry of the food into the duodenum, pancreas release pancreatic juice, and this pancreatic juice consists of some enzymes called trypsinogen, chymotrypsinogen, proelastase and procarboxypeptidase. So all these are in inactive forms and they should be converted into active forms. Enteropeptidase enzyme, also called as enterokinase, secreted by the intestinal juice, activates this trypsinogen enzyme into active trypsin. So trypsinogen with the help of enterokinase is transformed into active trypsin. So this trypsin then acts on the proteins and peptides and hydrolyzes them into peptides and dipeptides. So the trypsin is an autocatalytic enzyme, and after formation of some amount of trypsin, it undergoes autocatalytic reaction and transforms the remaining trypsinogen into active trypsin and acts on the proteins and peptides. So this trypsin is endopeptidase and it attacks on carboxyl and of basic amino acids, especially like arginine and lysine. And the trypsin also activates the remaining inactive enzymes like chymotrypsinogen. Chymotrypsinogen is activated by trypsin into chymotrypsin. So this reaction takes place at the pH 7 to 9, Chymotrypsin acts on the proteins and peptides and break down the proteins and peptides into polypeptides and dipeptides. Chymotrypsin has a dual role. This chymotrypsin attacks on the carboxyl end of aromatic amino acids like tyrosine, tryptophan, and phenylalanine. This chymotrypsin also acts as a milk coagulating enzyme. So renin is absent in adults. Renin enzyme is absent in adult. It is present only in infants. We have studied that it is present only in infants and calves. So in adults, the milk is digested with the help of chymotrypsin enzyme. It causes the curdling of milk or coagulation of milk. So the other enzymes present in pancreatic juice is proelastase enzyme, and this proelastase enzyme is converted to active elastase with the help of trypsin. Elastase enzyme attacks the bonds next to small amino acids like alanine, glycine, and serine. And next is the carboxypeptidase. Another enzyme is procarboxypeptidase, and this procarboxypeptidase is also activated by trypsin enzyme to active carboxypeptidase. As already said, this carboxypeptidase enzyme, it cleaves the terminal amino acid at the carboxyl end. The succus entericus consists of, succus entericus is the intestinal juice, and it contains two enzymes, that is aminopeptidase and dipeptidase enzymes. Aminopeptidase enzyme, it breaks a polypeptide chain at the terminal amino acid near the amino group. Dipeptidase is another enzyme which splits the dipeptides into polypeptides and dipeptides into amino acids. Polypeptides are acted upon by carboxypeptidase and they are hydrolyzed to dipeptides and amino acids under pH 7 to 9. Polypeptides are converted to dipeptides and amino acids with the help of aminopeptidase enzyme. Dipeptides are transformed to amino acids. Dipeptidase enzyme, it will split the dipeptides into amino acids. The end of protein digestion, several proteolytic enzymes, they break down the proteins into amino acids. The proteases include pepsin, renin, trypsin, chymotrypsin, elastase, carboxypeptidase, aminopeptidase, and dipeptidase. Now let us study about lipids. Animal and plant food contains lipids in the form of fats or oils. Phospholipids and cholesterol are also included in lipids. Lipids are chemically triacylglycerols or triglycerides. These lipids are needed in our daily diet, about 60 to 150 grams. Lipids are formed by joining of three fatty acids with a glycerol molecule by ester bonds. So this is a triglyceride. This is glycerol and fatty acids. Three fatty acids are there and they are joined by ester linkages. These lipids are digested by lipase enzymes. Now let us see how lipids are digested in various parts of the digestive system. Digestion in oral cavity. Mammals do not contain lipase enzyme in the saliva, hence lipid digestion does not take place in the oral cavity. However, lingual lipase is secreted in man, and this enzyme is secreted by the Ebner's glands present on the dorsal surface of the tongue. This enzyme is active only at the pH 4.5 to 5.4. But the oral cavity is not suitable for the action of lingual lipase. The food is swallowed and enters into the stomach through esophagus. In the stomach region, the heat of the stomach and also the peristaltic movements of the stomach make the bulk fats into they make the bulk fats so that they are easily, the turning movements make the bulky fat so that they are easily digestible. Lipid digestion occurs in the stomach region with the help of lingual lipase, as the stomach is acidic in nature. Both lingual lipase and gastric lipase are together called as acid lipases. These acid lipases, they help in lipid digestion in the stomach region, and about 30% of digestion occurs in the adult. In neonates, 50% of digestion occurs with the help of these both acid lipases. Neonates' diet consists mainly of milk, and this milk is digested with the help of these acid lipases. And acid lipases, they do not require any emulsification, they do not require the help of bile salts or co-lipase enzyme. They can act on the fats present in the milk, and they break down them into glycerol and small chain fatty acids. Next, the food enters into the acidic chyme enters into the intestine, and in the intestine it is mixed with bile, pancreatic juice and intestinal juice. Bile juice has no enzymes, but the bile salts, namely sodium glycocholate and sodium taurocholate, play a very prominent role in the digestion of lipids. They facilitate digestion by a process called emulsification. Fats are very large molecules with a very high molecular weight. So these molecules are made into smaller ones with the help of bile salts. Fat molecules or lipids, they are hydrophobic in nature, whereas lipase is hydrophilic. Hence, lipid digestion becomes difficult. Bile salts help in this situation, as they are both amphipathic and they are both hydrophilic and hydrophobic regions. So the bile salts, they bind to the fat droplets and they bind to the surface of the smaller fat droplets, and now the lipase can act upon such emulsified fat droplets, and the lipase will show its action and breaks, and these are easily broken down. Lipases are present in the pancreatic enzyme as well as the intestinal juice also. Lipases are present in pancreatic juice as well as intestinal juice. Pancreatic lipase cleaves the ester lipases at positions 1 and 3 of triacylglycerols and leaves the second linkage intact. So the co-lipase enzyme, it enhances the activity of pancreatic lipase enzyme. Intestinal phospholipase as well as pancreatic phospholipase A2, both these phospholipases act on the phospholipids in the presence of calcium ions and break down into glycerol, fatty acids, phosphoric acid and choline. Lipids are broken down to monoacylglycerol and fatty acid. So lipids are chemically triacylglycerols. They cannot be digested with the help of lipase enzyme. So bile salts first emulsify the lipids. They are turned into emulsified lipids with the help of bile salts, and later the lipase enzyme, the help of co-lipase and phospholipases, all the emulsified lipids are transformed to monoglycerols and fatty acids, and this reaction occurs at pH 8. So this is triacylglycerol. On acid hydrolysis, fatty acids is formed, and this is the glycerol molecule. Just a quick recap. Digestion, the definition of digestion is the process in which complex macromolecules are broken down into simpler absorbable forms is called digestion. There are two types of digestion: intracellular digestion and extracellular digestion. Intracellular occurs within the cell. Examples: amoeba, sponges and coelentrates. Extracellular digestion occurs in higher animals. And digestion of the end products of carbohydrate digestion or monosaccharides. Polysaccharides are broken down to monosaccharides, which include glucose, galactose and maltose. The protein digestion occurs with the help of proteases. Proteases include pepsin, renin, trypsin, chymotrypsin, elastase, carboxypeptidase, aminopeptidase and dipeptidase. All these enzymes, they act on proteins, and the end products are amino acids. And in the lipid digestion, lipase enzyme plays major role, and this lipase, the lipids are converted. The end products of the lipid digestion are monoacylglycerol and fatty